The overall goal of this method is to purify herpes simplex virus type 1 DNA from infected cells for the proteomic analysis of viral genome-associated proteins.
This method provides key insights into the involvement of cellular factors in herpes virus infection.
The main advantage of this technique is that it can be adapted to directly purify different populations of viral DNA from infected cells, allowing for the analysis of multiple aspects of infection.
Begin this procedure with culturing of MRC5 cells as described in the text protocol. Then, dilute HSV1 in cold tris buffered saline or TBS. For the following steps, it is important to turn off the blower in the tissue culture hood to prevent cells from drying out. Use a pipette to transfer the growth medium from the MRC5 cells to a sterile bottle to add back to the cells after infection. Infect the cells by adding 7 milliliters of diluted virus to the cells. Rock the cells every 10 minutes for 1 hour at room temperature. Following adsorption, aspirate the inoculum, rinse the cells with 50 milliliters of room temperature TBS, and replace the original growth medium. Then, incubate the cells at 37 degrees Celsius in the presence of 5% carbon dioxide. After the onset of viral DNA replication, around 4 hours post-infection, label replicating viral DNA. To do so, dilute EDC in 1 milliliter of growth medium and add it to the cell culture medium of infected cells for 2 to 4 hours.